Ahora. So, good afternoon and welcome to a new SEM webinar. Today, we have a seminar from the Developmental Biology and Genome Modification Group coordinated by Berta Alsina. Thank you very much, Berta, for organizing this webinar. And when you want, you can introduce our speaker today.
Okay, thank you very much, Monica. So today we have Sergio Casas Tinto. He's Científico Titular and he leads the group of modelos de enfermedades humanas en Drosophila. He has been working in Drosophila as a model system to investigate several human diseases. So he first did a PhD in la Universitat Complutense de Madrid. In 2006, he was a PhD already. And then went for a postdoc in the University of Texas. And there he started also using Drosophila to work on some human disease, neurodegenerative diseases by generating Drosophila models of prion proteins and also Alzheimer's disease. And then he came back to Spain, to CENIO, working on cell competition, which is a very important process for regulate the growth of the organs and also tumorigenesis. And then moved to Instituto Cajal, where he started a little bit working also in glioblastoma. And then there he got a Ramón y Cajal contract where he started leading his own group. And then he got a position as Científico Titular. And recently, he moved from Instituto Cajal here in the new Institute of Investigación de Enfermedades Raras del CSIC. And well, I'm very pleased that he can explain today all his new research, which is very interesting, and you will see it. Thank you very much, Sergio, for accepting this invitation.
Well, thank you, Berta, and thank you to all of you for this invitation. So I'm going to put my presentation, which I will try to convince you that cell-to-cell communication is important for tumor progression. So can you see the screen?
Yes.
Yes, good. I'm going to put the laser. Okay. So my idea today is to explain what we have been doing during the last years about cell-to-cell communication in the context of a brain tumor, in particular a glioblastoma. And we use different models, different animal models, but today I'm going to explain you the relevant results that we have obtained using mainly Drosophila melanogaster. We will discuss the advantages and the limitations of the model, of course, and the results that we have obtained. So basically, what we study is shown in this slide, which is the glioma cells are these cells in red here. And it was described in the 70s that glioma samples show something named perineuronal nest, which were these structures that in the patients you could find in the brain and were basically expansions of the glioblastoma that were surrounding groups of neurons. So all this idea started with the concept that the glioma cells grow and compartmentalize the brain and isolate neurons. And we wanted to know if there is any relation, any communication between the glioma and the neurons. So a little bit of description of what a glioblastoma is the most aggressive and primary brain tumor. It can be removed by surgery, but it has an infiltrative nature, which means that the glioma cells are going to grow, expand, and infiltrate through the brain, and it is mixed with the healthy cells, with the neurons and the other glial cells. Almost 100% of the patients after the surgical removal have a recurrence. That means that the glioma appears again. And if you see this image, this is the solid face of the tumor, but out of this solid face, you can have a huge mass of infiltrative tumor that is going to grow silent through the brain. The median survival of these patients is very low. It's about 16 months, and less than 10%, I would say even 5% of the patients survive after 5 years. So it's a very aggressive tumor, and it is lethal. If you pay attention to the symptoms of these patients, you can detect that they show headache, nausea, vomiting, drowsiness, and even after the surgical removal, these symptoms say and they increase. If all these symptoms, if you don't know that these patients have a glioblastoma, you may think that they have a neurodegenerative disease, because some of these symptoms are typical of a neurodegenerative process. So let's say about 10 years ago, we made the following question in the lab, which is basically what I want to demonstrate today, is that a glioma also causes a neurodegeneration, and that the neurodegeneration is relevant for the progression of the disease. So today I would try to show you some messages, some molecules that travel from the glioma to the neurons, or from the neurons to the glioma, and that at the end coordinate the cellular status of the tumor and the healthy tissue, and that depending on the physiological status of these two cells, the patient is going to have a different result. So glioblastoma has been very well classified genetically. This panel shows the most typical mutations that people has found in the glioblastoma patients, but in particular, EGFR and the PI3K pathway, either the PI3K enzyme itself or the P10 repressor, these are the most frequent mutations in the patients. All the other mutations are also relevant, but today we are going to talk about these two pathways, PI3K and EGFR. And actually, the model that we use in Drosophila is based on the expression of constitutively active forms of EGFR and PI3K. This model was done in 2009 by the group of René Reed and John B. Thomas, and they demonstrate very beautifully that Drosophila glial cells reproduce all the signaling cascade that is found in the patients, and that the activation of these two pathways will trigger the production of glioblastoma in the fly. This is the normal brain of a Drosophila larvae, and this is the brain of a Drosophila larvae after the induction of a glioblastoma with these genetic modifications, EGFR and PI3K. But don't forget that the interest of my laboratory is to know if the growth of the glioma affects the neighboring neurons. So to start with, and to know if the model is correct to reproduce this disease, it was not only the genetics, also the cellular equivalence of glial cells in Drosophila and mammals is well conserved, and there is literature demonstrated that all the cell types in the mammals and the functions are conserved in Drosophila, and they have equivalent cell types in the fly than the ones that we found in the mammals. So I always like to start the seminars just doing some spoiler. So this is what we have done, and it will be easier to follow the presentation if I explain now what we have found. I will describe today how glioma cells, they form filopodia that will facilitate the interaction with the neurons, and that in this interaction, in this space, which is very close, the neuron and the glioma cells, they produce these TMs, which are tumor microtubes, which are basically filopodia. They will exchange a number of signals, including Wnt pathway, which in Drosophila is called wingless, the IMPL2 and the insulin receptor, and Eiger and Grindelwald, which are the TNF-alpha in mammals. These are three examples of three signals that the receptor is in one cell type and the ligand is in the other cell type. And the production of the ligand is going to change the signaling in the neighboring cell. So to start with the tumor microtubes, in 2015, this publication by the group of Frank Winkler, they describe how human glioblastoma cells in a xenograph, so put in the brain of a mice, they record the growth of this glioblastoma for 60 days. And you can see here how the green signal is the tumoral signal, and they produce this filopodia that in this paper were described as actin-based filopodia. They call them tumor microtubes. Just reading this paper and the literature, we found that the description of the tumor microtubes or the glial network, they are relevant for invasion and proliferation of the glioblastoma, and they help to interconnect long distances in the brain. After radiotherapy, this network is very important for the repair of the glioma cells, and genetically, it is known that these tumor microtubes are dependent on the expression of GAP43, which is called IGLU in Drosophila. So reading the literature, this actin-based filopodia resemble the cytonyms already described in epithelial cells in flies. And in the 2000s, the cytonyms were described as cellular projections that in the epithelial cells contribute to the signaling between these different cell types. Those are also actin-based filopodia, and they were very similar to these novel tumor microtubes. So I tell you this because we took advantage of the tools that were done for the cytonyms. This UAS-IHOG was a marker to visualize the cytonyms. So we decided to put this marker in the brain of a fly with a glioblastoma. So this is the brain of the larvae, and we are going to pay attention to these hemispheres here. And if you see here, this is the signal of the cytonyms or of the filopodia in a normal brain, and this is the tumoral network that is formed after the induction of a glioma. So not only this tool is helpful to mark the filopodia in the brain, in the glial cells of the brain, but also they can show how the glioma condition in Drosophila also forms a superstructure of perineuronal nest. Well, to be sure that these are the perineuronal nest, we did a double staining. This is the membrane of the glioma, and this is a specific staining for neurons in Drosophila. And you can see how in these spaces that the glioma is generating, we can find neurons inside. So in the confocal microscopy, we could detect that the glioma is organizing some sort of a structure around neurons. If we move to the electron microscopy to have a little bit more of resolution, you can see in magenta the glial cells, and in blue the neurons, and in a control sample this is the normal scenario. But when we produce a glioma, we found not only an enlargement of the glial cells, but also they generate this type of tumor microtubes in Drosophila. In a higher resolution, we could see how this membrane of the glioma cells is organized around the neurons, and in some cases we saw up to 7 layers of enwrapping of the glial cells around the neurons. Well, but at this point we had a limitation, is that we could only see the superstructure of the tumor microtubes of the glioma network. So we decided to take advantage of the genetics in the fly. I am not going to go into detail about these constructs, but with this genetic tool, we can generate individual clones within the tumor, individual cells within the tumor. So this is the brain of a fly is a magnification. These are tumoral cells, but these ones in particular are marked with GFP. And we could see how the cells in vivo were forming these projections. So we knew at this point that the fly model is capable of generating tumor microtubes, one structure that is similar to tumor microtubes. As I mentioned before, tumor microtubes were sensitive to the expression of GAP43. So we decided to compare a control sample, glioma samples in Drosophila, and glioma samples that were expressing an RNAi against GAP43. And as you can see here, the formation of the perineuronal nest disappears if these cells, if this glioma in the fly do not have GAP43. In addition, we found that the lethality that is caused by the generation of a glioma is rescued by the expression of GAP43 RNAi. So we can conclude that GAP43 is required for the formation of the tumor microtube network, and that is relevant for the survival of the animal. Well, we know that there is a tumor microtube structure and that there are neurons within the perineuronal nest, but that does not mean that the glioma cell is interacting with a neuron. So we decided to do one experiment, which is called GRASP, which is basically the reconstitution of a GFP molecule. We express half GFP in the glioma cells, the other half of GFP in the neuron. If these two cells are very, very close together at a synaptic distance, which is called, we will see green, only if they are in contact. So when we use this reporter in the fly, we could see how control samples, they of course have some interaction between glial cells and the neurons, and it's the signal that you can see in this panel. But when we induce a glioma, the interaction is super enhanced. There is much more interaction between the glioma cells and the neurons. If you remember, these tumors that I was mentioning, they depend on the PI3K and EGFR signaling. And actually, we think that the formation of these tumor microtubes depends on the signaling of EGFR. But this is another story that we will go through it in another occasion. But if you see, these are the human cells, and we are comparing how they move. The ones on the left have higher signaling of EGFR. The ones on the right are also glioblastoma cells, but these ones do not have EGFR, upregulated EGFR signaling. If you pay attention, these ones on the left, they send filopodia, they send tumor microtubes, and they move based on a filopodia expansion. These ones on the right, they have an amoeboid movement, and they do not send the same filopodia. They just do like an amoeboid movement. So this is why we think that EGFR is relevant for the formation of these tumor microtubes. And actually, here you can see in red one single tumoral cell that you can see that is producing this large tumor microtube, and the rest of the cells are neurons. So we saw how these EGFR positive glioblastoma cells interact with the neurons surrounding in the same tissue through the microtube, through the tumor microtubes. But anyway, let's get back to the Drosophila experiments. The question at this point was, what is the function of the tumor microtubes, and which molecular mechanisms, if any, depends on the formation of these tumor microtubes? So we thought, and we read different papers of glioblastoma, and there were a number of evidences. The tumor microtubes are similar to the cytonyms, and we know that the cytonyms in Drosophila carry different receptors and ligands, including Wnt pathway as one of the main pathways. We know that Wnt pathway, the marker of beta-catenin, is one of the markers that is used for the prognosis of the glioblastoma, and having a Wnt-positive glioblastoma has a poorer prognosis. So Wnt seems to be a good candidate to be mediated by the formation of these structures. So starting with this point, let me give you a very brief idea of what Wnt pathway is. In Drosophila, Wnt is called wingless, and wingless is going to interact with the Fritzl receptors. There are Fritzl and LRP. Today we're going to talk about Fritzl receptors. So when wingless binds to the receptor, there is a chain of reactions that will release the beta-catenin, which is called armadillo in Drosophila. So out of this diagram, please pay attention to the ligand, wingless, to the receptor, Fritzl, and to the effector, which is the beta-catenin, which is armadillo, that will go to the nuclei and then will trigger the corresponding signaling. So starting with the ligand, we use a specific antibody that is going to mark wingless protein. And we use this is a magnification of the brain. We use wingless, and we saw that wingless has more or less an homogeneous distribution through the brain. But when we induce a glioma, you can see here the membrane of the glioma, and you can see wingless protein. It is accumulated in the membrane of the glioblastoma cells. And I say accumulated because wingless is not upregulated. There is no more protein, there is no more transcript in these samples. It's only moving from one tissue to another one. If this is the ligand that is accumulated in the membrane of the glioma cells, we also wanted to study the receptor. So this is the specific antibody for Fritzl1. This is the membrane of the glial cells, and we found a distribution through the brain. But when we induce a glioma, we could find that Fritzl1 was preferentially accumulated in the membrane that is forming the perineuronal nest. And here are the quantifications of Fritzl signal divided by the intensity of the membrane. So there is more receptor in the perineuronal nest when we induce a glioma. If we RNAi Fritzl, the signal is gone. And if we RNAi GAP43, so we reduce the formation of the network, but also we lose the accumulation of the receptor. In addition, we also did a survival assay, and we found that the lethality caused by the glioma is also rescued by the RNAi of the receptor Fritzl in the glioma. So at this point, we could say that Fritzl receptor in the glioma cells is relevant for the survival. So to determine, we know that the tumor microtubes is contacting the neurons, and we know that there is ligand and that there is receptor, but that doesn't mean that the ligand and the receptor are interacting. So we did an experiment of proximity ligation assay, which basically will help you to see two proteins that are very close together. And if you have any questions, we can go later through this protocol. But basically, if the proteins are very close, they have specific antibody with a chain of nucleotides that if they are close enough, we can do a PCR in the tissue and we will see signal when these two proteins are very close together. And we found that the interaction between Fritzl and wingless is 5 times more in glioma samples than in control samples. But having a ligand that is interacting with a receptor does not mean that they are signaling. So we use all the available reporters of the pathway, which in this case was 5 of them. These are different reporters of the wingless pathway. And we found that in all cases, this is the membrane of the glioma, and this is the signal of the reporter. In all cases, all the reporters were active in the glial cells, in glioma cells. But very importantly, the signal was gone, disappear from the neurons that were neighboring the tumoral cells. So Wnt signaling was imbalanced, was more in the glioma, but disappeared from the neurons. So all this molecular interaction, does it have any relevance for the progression of a tumor? We decided first to study what happens to the tumor when we modify these signals. So this is a magnification with a specific antibody to mark the glial cells, or glioma in this case, and we could see how there is an increase in the number of glial cells when we produce a glioma. There is a significant reduction when we produce a glioma knocking down Fritzl, so the receptor of the Wnt signaling is required for the increase of glioma cell number. And also, if we knock down GAP43, we also found that the increase in the number of glial cells, of glioma cells, also requires the formation of the tumor microtubes. But well, this is correct for the tumor, but how about the neurons, the neighboring neurons? We found that the neurons that are growing, well, these are neuromuscular junctions, and we use this tissue because the soma of the neurons is in the head of the fly, but the projection and the contacts, the synapses of these neuromuscular junctions are individual. There is one per muscle, so we can count the number of synapses. And this is relevant because reducing the number of synapses is one of the first symptoms of a neurodegenerative situation. So we use this particular structure to measure neurodegeneration. This is the normal structure of a neuromuscular junction, and this is the structure when we induce a glioma. And there are several things. There is clearly a change in the morphology of the neuron, of the synaptic buttons. There is clearly an expansion of the glial membrane, which is in red compared with the controls. And when we count the green dots, which are synapses, we can count the number of synapses per neuron. We see a significant reduction in the number of synapses. In addition, we also found that the control animals, they have a behavior which basically they climb through the tube, and when we induce a glioma, 12 days after the induction, we see this situation. They walk very slowly, if they walk, because in many cases they stay in the bottom of the tube, having some sort of seizures, but of course a limitation in the movement, which is also very typical of a neurodegenerative situation. Well, so far I have shown you that glioma cells are sending these tumor microtubes, these projections that accumulate the receptor, and we propose that this is going to capture wingless. So if the receptor is important to compete for the ligand for wingless, we thought that maybe if we only generate transgenic animals with additional Fritzl receptor, they could compete with the glioma. So if the competition for wingless is relevant, then if we put extra receptor in the neurons, then the neurons should be able to compete and maybe to avoid the neurodegeneration. So when we did these experiments, here you can see a glioma, and this is the size of one of the hemispheres with the expansion of the tumor. And here you can see a glioma, which is genetically complete, but the neurons in front of this glioma, they upregulate the receptor. And as you can see, this is the same magnification. The tumor does not grow. So there is something that modifying the neuron, you can prevent the expansion, or you can delay the growth of the glioblastoma. Well, the tumor is growing less, but how about the neurons? Are the neurons healthier now? So we counted, again, the number of synapses in different scenarios, a control, a glioma, and then a glioma in a brain where the neurons are expressing, overexpressing the receptor. And the reduction in the number of synapses that occurs due to the glioma is rescued just by adding the receptor to the neurons. So yes, the neurons are healthier when they upregulate the signaling of Wnt pathway. In addition to this, we did different genetic modifications. So again, we counted the number of synapses in control, glioma, a glioma that does not express the free cell receptor. So if the neurodegeneration is mediated by the Wnt pathway, now we remove the receptor from the glioma, and the neurons are better. They are significantly better. They have more number of synapses. A glioma that cannot generate the network, this is gap 43 in flight, sorry for the typo. If the glioma cannot generate the network, also the neurons have a normal number of synapses, but here we had the same question. Okay, we can rescue the number of synapses, but the type of glioma that you are generating is incomplete. So genetically, the glioma is PI3K and EGFR, fine. But in all these cases, for example, when we knock down free cell, we are reducing free cell signaling. Maybe these glioma cells are not glioma anymore. So we did a genetic combination that we generate a glioma, we knock down free cell, but we express a constitutively active form of armadillo, of beta-catenin. So this glioma signals for Wnt pathway is genetically a glioma, but does not have the receptor. And the number of synapses is significantly larger than in the glioma alone. So it's clear that the receptor is mediating the neurodegeneration of the neurons, even though this glioma is signaling for all the required signals, the lack of the receptor is enough to rescue the number of synapses. So these combinations, we wanted to study both the growth of the tumor and the neurodegeneration. So regarding the tumor, there is one very important question. Who is producing winglets? So far, we don't know. We know that there is more winglets accumulated on the membrane of the glioma. We know that there is no upregulation, there is no more protein, there is no more transcript, but we don't know which is the source of winglets. So we did a genetic experiment. We generate a control samples and the membranes. We generate a glioma and the membrane of the tumor. We generate a glioma and we knock down winglets in glioma cells. And as you can see here, winglets is not necessary in the glioma cells to develop a glioblastoma. So the source of winglets is not the tumoral cell itself. And here I show the combination that I was explaining to demonstrate that this glioma without free cell, but signaling through armadillos, through beta-catenin, fully develops a glioma comparable to the normal ones. So very important, winglets is not generated in glioma cells. So the only option that we could think about is that winglets is coming from the neurons. If to validate this hypothesis, the question is, what if we generate a glioma, but we block the access to winglets in the neuron? So we took advantage of one tool, which is a GPI anchor version of winglets. If you pay attention to this diagram here, the proposal is that the glioma is going to accumulate the receptor and is going to take winglets from the neighboring neurons. Now we are going to use and these winglets, once interacts with the receptor, will trigger the signaling of armadillo. Now we are going to use a GPI anchor version of winglets, which is anchored to the membrane. So it is impossible, or at least much more difficult, to retrieve this ligand from the neurons. So when we did this, you can see here a control sample. This is the expansion of the glioma in a fully in a genetically generated glioma. And this is a glioma that in front of this glioma, the neurons have this anchor form of winglets, and the glioma does not grow. If you compare, this is the whole hemisphere. This is at the same magnification, but you cannot fit the whole hemisphere here. And this is again an hemisphere of the brain that fits perfectly, and it's very similar to the controls. And remember, the only modification here is that we modify the access to the ligand in the neighboring neurons. So in this case, we quantified this and we found that the gliomas cannot grow if they cannot get winglets from the neurons. So far I have explained to you that glioma is going to form these tumor microtubes that will accumulate the receptor. This will help to deplete winglets from the neurons. Actually, this is a process that Marta Portela in my laboratory named vampirization. Basically, the glioma vampirize the neuron to take winglets. This will promote proliferation and survival, and in consequence, will reduce the number of synapses and will cause a neurodegeneration. This story is a little bit longer, but I'm not going to go into details, but I'm going to at least give you a very brief concept of this. PI3K and EGFR produce a remodeling of the cytoskeleton that will trigger the generation of tumor microtubes, will facilitate winglet signaling. This also upregulates the GenK pathway in the glioma cells, that this promotes the production of matrix metalloproteases, which are enzymes, proteins that are secreted to the extracellular space and contribute to digest the extracellular matrix. This facilitates the expansion of new tumor microtubes that again will trigger novel winglet signaling and again will generate this positive feedback loop that potentiates the expansion of these glioma cells. So this is one example of how a signal generated in the neuron goes to the glia and signals into the tumor to produce the expansion of the tumor. But in the lab, by that time, it was Patricia Jarabo in my laboratory, and she wanted to know if there is something that generated in the glioma goes to the neuron to change the physiology or the function of the neuron. So we know that from the neuron, winglets is going to go to the glioma, but Patricia wanted to know if from the glioma, something goes to the neuron to change the activity of the neurons. So I am going to go very briefly through this idea. In the literature, we found that IMPL2 in humans is insulin growth factor binding protein 7. It's a molecule that has been related to the insulin receptor. This is an antagonist of the insulin receptor signal, and when it is present, it blocks the signal of the insulin receptor. We propose this molecule as something that could be produced in the glioma. Actually, in the databases, there was a lot of information showing that in glioblastoma samples, there was a high expression of IGF-BP7, and that the upregulation of this molecule was related to a poor prognosis, a shorter lifespan of the patients with glioblastoma. In addition, if we want to propose this molecule coming from the glioma and traveling to the neurons to block insulin receptor, I have to say that insulin receptor signaling is super relevant for the formation of synapses in the neurons. So at this point, this was a good candidate to study if it was generated in the glioma and affecting the neighboring neurons. We found an upregulation also in Drosophila of the expression of IMPL2, and we use a specific fluorescent reporter. This is control sample, the reporter is in green. Glioma samples, the green overlaps with the red signal, which is the glioma membrane. And if we knock down IMPL2, the reporter disappears. So it looks very fine that IMPL2 is upregulated in glioma samples, particularly in glioma cells. Also, we did a little bit of genetic interactions. We block the GenK pathway or we reduce the number of tumor microtubes. And in all cases, GenK pathway and the formation of the tumor microtubes is necessary for the upregulation of IMPL2. Well, let's go for the mechanism of IMPL2. IMPL2 should block the signaling after the insulin receptor. And this is a simplification of the pathway, but there are, among all these players, please pay attention to Reb. We know that when we induce a glioma, we find a significant reduction in the number of synapses. And we wanted to know if this can be mediated by IMPL2. So we counted the number of synapses in control samples, in glioma samples, and now in glioma, but knocking down IMPL2. And we observe that there is a rescue of the number of synapses. Moreover, we generate the upregulation of IMPL2 in wild-type glial cells. And upregulating IMPL2 in normal glial cells is sufficient to reduce the number of synapses in the neighboring neurons. So IMPL2 is necessary and sufficient to trigger a reduction in the number of synapses. If our hypothesis is truth, it should be enough to upregulate insulin pathway in the neurons to restore all to a normal condition. So we generated a glioma in glial cells, and in the neurons, we upregulate the expression of the gene Reb, which is this one here. When we did this modification, we can see how the glioma causes a reduction in the number of synapses. And if we upregulate insulin pathway in the neurons, we can rescue the number of synapses. So insulin pathway is the target of this molecule that is produced in the tumor, but causes an effect in the neighboring neurons. We use a reporter of the pathway, which is very contraintuitive because this one is going to give a signal when the pathway is down, when the pathway is off. So we found this is the normal signal. When you find a nuclear signal, means that the insulin pathway is off, and you can see these are neurons in a glioma sample. You can see how the insulin pathway is off. When we generate a glioma, but we knock down IMPL2, we rescue the normal situation. So IMPL2 was mediating the reduction of the insulin pathway in the neighboring neurons. While doing these experiments, we found that mitochondria, and this is a very complex slide, but I'm going to go only to these images here on the right. Mitochondria is a marker for mitochondria. And we found that in the control sample, glioma sample, we found an accumulation of mitochondria, so mitochondria, in the projections of the neurons. So it's a symptom that something is not okay, that the neuron is undergoing a degenerative process when you find mitochondria accumulated in the axons or in the soma. And also we found that when we induce a glioma, but we upregulate Reb in the neurons, we rescue, again, a phenotype similar to the control. So the accumulation of mitochondria caused by the glioma is rescued if you activate insulin receptor in the neurons. In addition, we also wanted to check in the electron microscopy, which has better resolution for these experiments. This is a normal mitochondria in a neuron in a control sample. This is a mitochondria in a neuron in a glioma brain. And this is the mitochondria in a neuron of a glioma sample, but these neurons are rescued with the activation of the pathway. So all these observations indicate that there is a molecule coming from the tumor that goes to the neuron and causes a change in the neuron. But at the end of the day, is this useful? I mean, does something change in the animal? Does it live better? Does it live longer? So we did a survival experiment, and you can see how control animals are gray. In black, you can see the glioma and that they die around day 20. And in blue, these are animals with a glioma, but the neurons are protected by the upregulation of insulin receptor. So what is the message of all this story? Is that glioma cells and neurons exchange signals. There are signals going from one side to the other. And these signals are integrated in a very complex system that at the end will trigger the physiological status of the neuron and of the glioma cells. Now we have to add IMPL2 as a pathway for the insulin, as a molecule for the insulin receptor pathway that will change the status of the neuron, and this molecule is generated in the glioma. So this part is to convince you that there is a communication, and I am going to use the last 5-6 minutes to explain to you why this is relevant and what are we doing now. Everything in the most recent project began with this idea. This is the survival of glioma patients at different ages, but as you can see, it is not possible to predict if you are going to live 1 year, 2 years, 3, 4, or 5 years. So this happens in many, many diseases, especially those related to the nervous system, that it is very difficult to predict the survival of the patient. As the same thing happens with aging, not only the survival, also the quality of the function. And we wanted to propose that maybe the status of the brain, the health of the brain, is relevant to resist the growth of a tumor or any other neurodegenerative disease. We decided to call to this concept brain fitness, which is basically the ability to the brain cells to resist the impact of an aggression, in this case, a glioma. So we wanted to study what factors can change the brain fitness, the ability of your cells to survive. And the proposal is that all our brains have passed the developmental filter. So all of us, we are functional, but we don't know how our brain can handle different aggressions, and we don't know how to predict that. We propose that there are many studies trying to relate epidemiological situations or habits with the progression of a glioblastoma, but they did not exert a very good result. So we propose that there is something that is inherited from the parents that will change the fitness of the brain in the progeny. So what we did is to induce stress as a modulator of brain fitness. But we induce stress in the mothers, in the parental line, and we analyze the progeny, the F1 generation. We have used 2 different stresses, light, permanent light, and high-fat diet with coconut oil, okay? Basically, we stress the mothers, and then we cross them with normal parents, and then we analyze the next generation, which is not exposed to any stress. First thing we did is we stressed for 7 days the females of Drosophila, and we analyzed the ovaries, and using a reporter of mitochondrial stress, which when there is a stress, it becomes red, we found that under normal conditions, there is this basal situation, but when we expose the animals to permanent light, there is clearly a mitochondrial stress, which is quantified here. Then we cross these animals and we analyze the next generation, and we dissect the brain of the next generation. And we found, using the same marker, that the progeny of the females that were stressed show symptoms of mitochondrial stress in the brain of this F1 generation. So first of all, we wanted to propose that there are inheritable factors that are caused by maternal stress, but you can see them in the next generation. And the first question was, is this relevant for the normal development of the flies? So we analyzed different parameters of these animals. We analyzed the generation of the glial membrane, and there were no significant changes. This is a particular point, but we require larger numbers. But in general, there is an impact in the development of the glial cells. There is an impact in the lifespan, so the next generation lives normally, the males and the females. But when we when we analyze in details the females that were coming from a stressed mothers, they have a slight shortening in the lifespan. At this point, we didn't know if this was relevant, but it was reproducible. So always these females live a little bit less than the rest of the females or than any of the males. The males seem to be resistant to the inheritable stress. So now we combine this situation of you are born from a stressed mother, and we induce a glioma in these animals. And we found that in the males, there is no significant difference, but in the females, when we induce a glioma, there is, this is the normal, the survival of the glioma, and this is the survival of the animals with the glioma, but with stress mothers. So the stress causes something that shortens the lifespan of these females when they have a glioma. And here there was like the starting point of this part. So basically we studied the progression of the tumor, and I'm going to go very quickly, but this is the membrane of the normal glial cells. When we induce a glioma, this is larger, but if we induce a glioma and the animals come from stressed mothers, both males and females, the glioma grows faster, okay? And this is the analysis 5 days of life. When we do this at 7, there are the differences in the glioma and glioma in a stress progeny disappear. So it's not that the tumor grows more, it only grows faster. Also, we analyzed the number of synapses, and we found that at 5 days, there are no differences in any of the samples, control, glioma, and glioma coming from stressed mothers. But at 7 days, we found a significant reduction of synapses in the neurons of animals that were coming from stressed mothers and had a glioma. So the glioma grows faster, and now the neurons lose synapses also faster. This proposal is always identical for males and females, but if you remember, the females were more sensitive in the lifespan, and this is something that we, at that point, we did not understand. The same situation with the number of glial cells. The glioma causes an increase in the number of glial cells, but if this is in a brain that comes from stressed mothers, the number of glial cells is larger in males and in females, and these differences, when it's at 7 days, they are equal. So indicating that the glioma grows faster, but it does not grow more, it only grows faster. So this is what we know at this point, is that at 5 days, the formation of tumor microtubes is larger of the glioma cells that are in an animal coming from a stressed mother, and that at 7 days, the number of glial cells and that the neurodegeneration is more aggressive. But in all cases, males and females behave very similar, which we did not understand because the lifespan of the females was significantly less than the one of the males. So we analyzed the brains of these animals. We did an RNA-seq, and I am going to go straight to the point. If we RNA-seq the heads of the flies that are born from stressed mothers, and we compare females and males, these are the controls, and these are the females coming from a stressed mother, males coming from a stressed mother. You can see how the number of specific changes in males and females is more or less the same in the controls, around 500–600. But if the animal comes from a stressed mother, there are more than 1,000 differentially expressed genes in females and only 300 genes differentially expressed in males. And this is a similar analysis, but all of them indicate that the females suffer transcriptional changes in the brain in a larger manner than the males. There are changes in both sides, but the females suffer more changes than the males, the different in the gene expression. In addition to this, we compare the genes that are changed in both cases. So all the genes that are shared by males and females exposed to light, these ones here, if we analyze them in detail, most of the genes change. Most of the genes change in the female in a larger manner. So all these expression changes indicate that the brain of the female is more sensitive to the light exposure. The brain of the female in the progeny is more sensitive than the brain of the male, and that there are more transcriptional changes. The nature of these changes is very heterogeneous, and there are signals which are relevant for synapse generation, for glioma progression, and we are right now studying these genes. But we wanted to know, one very quick question is, what is stress? It's something very difficult to define, and we decided to compare the RNA-seq of animals that are coming from light stress compared with the RNA-seq from animals that come from a different source of stress, in this case, high-fat diet. And we fed the parental line with high-fat diet, and we analyzed the progeny, and we found that now most of the changes now occur in the progeny, but in the males. So the situation is inverse. Now it looks like, depending on the source of stress, most of the changes now occur in the males that are the progeny of high-fat diet animals. So this indicates that the source of stress and the sex of the individual, there are combinations that will exert different results between males and females. So just to finish this, if we cross all this data, high-fat diet and light, and which are the conditions that always change. So no matter if you are male or female, if your parents were stressed with light or with high-fat diet, there are 128 genes that always change. And now we are analyzing which is the relevance of these 128 genes in what we call the brain fitness. As you see here, the identity of these 128 genes can be classified by these different gene ontologies, but still we are now analyzing which is the relevance. So to finish with, what I want to propose is that there is something that we want to call brain fitness that depends on the diet, on the exercise, on the sleep, or on the habits of the parents that generate the status of the brain. These ones are preconditioned as they come with the fact of being born, and these ones can be acquired in your daily life. But at the end, the brain will be capable of resisting or not to any pathological aggression or to aging, which is something that we are studying right now to see if there is any impact in aging. So all this has been done by a lot of people, and I would like to thank all of them, the people in the Cajal Institute and the people at the Instituto de Salud Carlos III, where we are located now. Also, I want to thank the confocal microscopy unit, which are super important for our projects, the funding agencies, and if you ever need to reach me, please reach me here. I will be very happy to answer to any of your messages, and thank you very much.
Okay, thank you very much, Sergio. Very interesting talk. So we will let some minutes, few minutes, in order that people can write the potential questions in the chat. And I don't know, Berta, if you want to start asking.
Questions. Yes, I have several ones. So yes, with the electron microscopy images in which you see these tubes, I see very well the tubes when there is in cell cultures, no, when the glial cell produces, but on the other, while there is not development that is surrounding the neurons, it looks like more kind of sheeting, no? Like
myelin. Looks like
myelin.
myelin.
It
Yes.
really looks like myelin.
It
No?
looks like myelin. The thing is that at the first sight, I thought it was myelin. Well, Drosophila does not have myelin, but it has a structure which is equivalent. Does not have the molecule myelin, but it generates an sheathing glia around the axon. I cannot discard the idea that glioma cells behave as oligodendrocytes. Actually, if you check on the single-cell RNA-seq, the identity of glioma cells is very heterogeneous. And if you want to select a group of them, they are like oligodendrocytes. So we have discussed a lot about the identity. What type of cell is a glioblastoma cell? And I don't know, but I would bet that this is any identity that you want to claim. Oligodendrocytes, also they have markers of astrocytes, so they have different identities. So probably this is a behavior similar to the oligodendrocytes, I agree. And this is what facilitates the contact with the healthy tissue.
And have you seen if then neural activity, I mean, neural activity probably is also reduced because you reduce synapses, no? But you could also think that it reduces synapses because you have less cells, less neurons. I don't know if there is cell death, but also it could be less activity because you are kind of...
Well, different things. The reduction of synapses is super early, and we have tried to block apoptosis in the neurons, and it does not improve in any way. Actually, we tried to see apoptosis, and you can see some of it, but it's not super significant. Actually, we think that the collapse of the nervous system is sufficient to kill the animal, so there is no time to induce a very massive apoptotic process. It is known that it changes the electrophysiological records of the neurons. We have not done it, but it is published that the electrophysiological recordings of the neurons is changed. We have seen synapses. I mean, there is reduction of synapses in the neurons, but also the glioblastoma interacts with the neurons through synaptic structures, which I didn't want to include it in the seminar, which was going to be very long. But the activity of the glioma is coupled with the activity of the neurons. So the proposal would be that the communication between both cell types alters both cell types. The activity of the nervous system, which I think that part of the symptoms of the glioblastoma patients come from the alteration of the nervous system, and this activity is going to feed the glioblastoma to expand because the activity is coupled with calcium waves in the glioma and proliferates more. So this is basically the idea that the communication couples both cell types, both of them change, but change in a direction that is a pathology.
And then, can I do another question? So regarding the signaling wing frizzle, the idea is that because you don't have more wingless or more frizzle, is that in normal conditions, wingless is in excess. So usually you have more wingless, and it's just that when you are touching this and you're closer, this wingless is activating more the glia, and then it's producing more proliferation. But if you're not producing more, at the end, wingless would be exhausted.
That's the idea. The idea is that the activity of the glioma taking wingless is depleting wingless in the neuron. And at the end, and we have done this experiment, you knock down wingless, you knock down the receptor in the neurons degenerate super quick, super fast.
Okay, so you have frizzle also in the neurons. I thought
yes. Normally you have. If you put extra, the neuron is more healthy. If you artificially, genetically, you knock it down, the neuron degenerates. So we know that the neuron depends on wingless signaling, and we think that at the moment that the glioma takes the ligand, the neuron is going to degenerate as a secondary effect. But I think that this secondary effect is super relevant for the outcome, for the survival of the animal. So it's not that secondary.
Okay. Do we have questions from?
So if you provide wingless,
We have
you can
tried.
see.
We have tried. The problem is that overexpression of wingless causes severe defects during development. So if you upregulate wingless, normally the animals are not viable, at least in my hands. So we have done this experiment in vitro. What we have done is we have first, we put glioma cells in a microfluidic plate, and we put picomoles of wingless in one particular source. So all the cells face to the wingless source. So glioma cells can detect wingless and can orientate and send the phylopodia to the wingless source. Besides that, if you co-culture human glioma cells and neurons, the neurons are going to get lost in the culture. So with time, it's going to be only glioblastoma. If you put additional wingless, the neurons live longer, not forever, but they double the time that they survive. So probably there are other signals. I don't think that this is only wingless and IMPL2. We have done things with hedgehog, with BMPs. There are many signals, but wingless is of relevance for sure. And the neuron survives longer if there is some extra wind in the culture. But I have not done that in vivo, as the animals are not viable.
Okay, thank you.
Thank you.
Okay.
And my question is regarding the translation to human glioblastomas. So now that you have identified some of these molecules, for example, the IGF, no? Do you think those are
Well, the distance between fly and human is very large, but in this case is probably the only case I am confident into giving it a try, because the pathways are very well conserved. Actually, these pathways were initially discovered in the fly, and they are conserved. What is the situation? A human is much more complex in the number of players. Wind ligands are dozens of them, and we have tested all of them, and only Wind3A and Wind5 work. These are the only two that work. The number of receptors in human are also larger, but we have not done the study yet. So we are working in particular inhibitors of Wind. The thing is that it's not clear if that is a good target, chemically speaking. I know that some laboratories are working in GAP43. So going one step back, going to the starting point of the problem, it is clear that the tumor microtubes are relevant for all the conditions. So if you block that, that would be a better strategy under my point of view. And actually, I didn't show it, but GAP43 is absolutely irrelevant for healthy glial cells. Only tumoral glia requires GAP43. So I think that GAP43 would be a better idea. But anyway, wingless, we have been doing things, but the problem is that this molecule in development is super important. So as a concept, it was very good to discover this cell-to-cell communication, but to select a particular target, I would go more for something that is structural, for the tumor microtubes, for example, because this tumor is called glioblastoma multiforme. What does it mean? That it has a number of subclones within the tumor, and molecularly, these subclones can be different. So one can be Wind dependent, but the other one can be hedgehog dependent and could be blind to Wind. And in the experience of the years, a lot of laboratories have tried to knock down PI3K, EGFR, as these are the primary mutations. But these tumors somehow get to adapt and find another signal that stimulates proliferation. So if I would have to start it now, I would go more for a structural thing. And the phylopodia are a good point, that you avoid the contact, the communication, and that is enough. We have one experiment, very beautiful experiment, where we knock down the production of matrix metalloproteases. So the tumor genetically is active, but it does not infiltrate. It infiltrates much slower. So, and the animals survive almost to the wild type. And it's only to stop the invasion, the infiltration. So there is one chemotherapy that works more or less, temozolomide. It's the only available chemotherapy in the market. It only expands 3 or 4 months the life of the patient, but temozolomide, you have to give certain amount of doses. If any of these modifications could expand the life of the patient, then you might be able to give temozolomide for a longer period of time, and maybe the combination of different strategies would give a better result. Because I think that for the glioblastoma, oncologists and neurologists have to get together to a point that one is going to try to stop the tumor, the other one is going to try to reinforce the neurons or the healthy tissue. When these groups get to work together, I think that this will improve a lot.
And for the, I think that the good thing of knowing maybe some factors that is not GAP43, it would be nice for the liquid biopsies, no? Because maybe you can have a prediction in just knowing some molecule that has gone into the...
Yep. It would be great to have a marker that can determine that you have a growing glioblastoma, because actually most of the patients, they find that they have a glioblastoma because they go to the clinic because they have a drowsiness, they have a problem in the speech or memory, and suddenly, oh, there is a tumor in the brain, because they are super silent until they get certain size. The limitation that I see is that most of these molecules are I mean, the tumor is not inventing anything new. They are using the regular signals, but they imbalance the equilibrium to their benefit. So it would be good, but of course there has to be a very fine study on each individual, each condition, each situation. Because one of the things we have found in the last experiments I have shown to you in flies, that if you are born from a stress situation, many of the molecules that change are exactly these ones: the ligand of Wnt, the receptor, the hedgehog, BMPs. So the same candidates that are used in this communication are some of the most sensitive to your personal condition. So I think that personalized medicine, which in my opinion is going to be the future, and I hope I will get to see it, but I don't know, that will bring an regular analysis of your personal levels of whatever we want to study, but has to be individualized. We cannot stay working in the generalization. Like, I did this clinical study with males of 35 years old. Okay, then females of 60 are also good for this study. No, each person, I think, that will require personalized medicine in a future. I don't know when that is going to come, but I think that that is going to happen.
Okay, I would like to ask about the last part where you see the sex differences, and also, well, I mean, you've been analyzing all the time RNA-seq, I guess, if I have understood. Have you tried to look at epigenetics?
Yes, actually, we have now a manuscript we are submitting a manuscript that describes what is the epigenetic modification that controls at least part of this. So when we saw the results, we thought that, one, there is something that is left in the egg that is going to change something in the animal. Second, there is an epigenetic modification that can affect the next generations. We found one epigenetic modification that should be published soon, but the thing is that this is the mechanism that we found, but I am sure it's not the only one, because among all the RNA-seq, we found a number of long non-coding RNAs. That is the project that we are doing right now, and that requires a lot of combinations. I'm very glad that artificial intelligence exists, because it can propose all sorts of combinations, but long non-coding RNAs is something that we have not studied enough yet. It's a very complex field, but super interesting, because in this case, seems to have the key of part of this situation. So, I mean, this is a field that has emerged in the last 10 years, the impact in the next generation of the activity of the parents, and there are positive things, negative things. So once we have the understanding of how all these inputs give a brain fitness, we will be able to predict, hopefully, how strong your brain is, which would be something good to know. Not only for a disease, I am super interested in aging. I would like to know why some people ages very good and why some people ages really, really bad. So I think that there has to be something which is not directly your lifestyle, which can also have an impact, but there is something else, because you cannot predict, okay, if you do this, and this, you're going to be good. That's not like that. So I think that there is something that is the inheritance that will come through epigenetic modifications.
Yeah.
Yeah,
Very interesting.
Your last part is very interesting, but it's a little bit, how you say, it's interesting, but at the same time, it's worrisome, no? In the sense that you think, okay,
I mean,
maybe I cannot change anything because I'm already have my brain with fitness, or really I can do something.
I would like to be positive, saying if you know it, then you can do something. So if you know that something is not ideal, maybe you can change it. So the information is important. Then anybody can do whatever they want with the information. But to have the opportunity of saying, look, according to the statistics, your brain might be weaker for a Parkinson's disease. So please pay attention to your substantia nigra, just in case. So go to review more often, or you're going to age very poorly. So we recommend you to do some exercise and things like that. So I don't have a clear idea on where this is going to take us, but it's a novel path and it's an exciting idea. So we will see how it goes.
Yeah, sure. Okay. So I think there is no question in the chat. So I think we can finish here the seminar. I would like to thank you, both of you, okay, for your nice presentation and your nice introduction. So take care and see you in the next webinar.
Thank you very much, Monica.
Bye.
Thank you very much, Sergio.